Hey guys, welcome back to That Bio Girl. I'm your host Belladonna, and today we're going to be talking about Unit 2, cell structure and function, and things like prokaryotic versus eukaryotic organisms, the endosymbiotic theory, the central dogma of biology, the endomembrane system, the inner parts of the cell and their functions, the cell membrane, cell size, water potential, and osmosis. So let's get started. So first, let's talk about the difference between prokaryotic and eukaryotic organisms. And let's start with prokaryotic ones. Prokaryotic organisms include bacteria, and they consist of one cell, which is why those cells are called unicellular. The different types of bacteria are eubacteria, which is the type of bacteria that lives in normal conditions, and archaebacteria, which live in extreme conditions. It is important to remember that prokaryotic organisms have no nucleus and no membrane-bound organelles, and instead of having a nucleus, they have a nucleoid region, which is in the shape of a large circle that stores their DNA. Additionally, they can sometimes contain extra DNA called a plasmid, which is also circular and can be transferred through thread-like structures known as pils. While they don't have a nucleus or membrane-bound organelles, they do have ribosomes, a cell membrane, genetic material, and a cytoplasm. Now remember when I talked about structural polysacs in episode 1? Well, most bacteria's cell walls are made of peptidoglycan, which is a structural polysac carbohydrate. But back to the last thing to remember, prokaryotic cells reproduce asexually, meaning that they just reproduce by making clones of themselves in a process called binary fission. On the other hand, eukaryotic organisms reproduce by meiosis and mitosis, which we will talk about later as well. And eukaryotic cells are much larger than prokaryotes and consist of protists, which are unicellular, and fungi, animals, and plants, which are all multicellular. And just a fun fact about animals and protists in particular are that they have cilia and flagella to aid in movement sometimes. But back to eukaryotic cells as a whole, eukaryotes have a true nucleus and membrane-bound organelles, so they are already so different from prokaryotic cells. And in terms of structure, fungi have a chitin cell wall, while plants have a cellulose cell wall, which again are both structural polysaccharides. Moreover, eukaryotes have ribosomes, a cell membrane, genetic material, and a cytoplasm, and their cells have compartmentalization, which means that its cells have organelles in which chemical reactions can happen in many places all at the same time, meaning that there is a higher surface area to volume ratio present, which is great for a cell's efficiency, which we'll talk about a lot towards the end of this video when we talk about cell size. Okay, now that we've talked about the individual differences between prokaryotic and eukaryotic cells, let's talk about what all cells have in common. All cells have genetic material, a cytoplasm, a cell membrane under the cell wall in plants or bacteria, and the ribosomes, which are non-membrane bound. Additionally, all cells maintain homeostasis, store and transmit information and transfer energy, and according to the cell theory, all organisms are made up of cells. Cells are the fundamental unit of life, and cells come from pre-existing cells. Now that we know about what they all have in common, let's talk about the endosymbiotic theory, which is a theory about how eukaryotes could have evolved from prokaryotes. Now, I will be honest with you, I personally don't believe that the idea of the endosymbiotic theory seems plausible because I'm a Christian, but I have to teach it to you because you certainly have to know it for the AP exam. So basically, this theory posits that the origin of the mitochondria, for instance, is that a free-living aerobic prokaryote was engulfed by an anaerobic, meaning not needing oxygen, cell through endocytosis, which we'll talk about later when we discuss the cell membrane and how it functions. Supposedly, the engulfed prokaryotic cell did not get digested by the engulfing cell. Rather, their relationship became mutually beneficial, and over time, the engulfed cell lost some of its independent functionality and became the mitochondria of the eukaryotic cell. As for chloroplasts, as the free-living photosynthetic prokaryote was engulfed by another prokaryotic cell through endocytosis, once again, and the engulfed prokaryotic cell did not get digested, but rather benefited from the endosymbiosis, which refers to this mutually beneficial relationship with one living thing living inside of another. Over time, the engulfed cell supposedly lost some of its independent functionality and became what we know today as the chloroplast. And while I don't agree with this theory, here's some evidence that biologists have found for the endosymbiotic theory that you should certainly know for this course. Both mitochondria and chloroplasts have a double membrane, which suggests that perhaps one thing did engulf another. Second, like prokaryotic cells, mitochondria and chloroplasts also have their own circular DNA, and they encode genetic information and can reproduce in a similar way to prokaryotes. And three, both chloroplasts and mitochondria contain their own ribosomes that synthesize proteins, just like prokaryotes. Now we are on to the inner workings of cells, so we just have to talk about the central dogma of biology, which states that DNA is used to create RNA, which is then used to create a protein. And this is how and why cells exist, which is why we're talking about it in relation to cells. Next, let's talk about the endomembrane system, which basically consists of the nuclear envelope or nuclear membrane and nucleus, the endoplasmic reticulum, the Golgi apparatus, the lysosomes, the vesicles, and the cell membrane. The reason why they are part of an endomembrane system is because these structures are all interconnected and made of membrane material. You see, they're all connected since to transport items between these parts, vesicles pinch off of the membrane material and travel from the nucleus with mRNA that is used to create proteins in the ribosomes, and those proteins go to the rough endoplasmic reticulum and then the Golgi apparatus to be modified and then out of the cell or inside the cell for a certain purpose via vesicle transportation once more. Now let's talk about the names of these organelles in the cell and their respective functions. And I know there are a lot of them, but stick with me. The different organelles, which are all membrane bound except the ribosome, are the cell membrane, the nucleus/nuclear envelope that envelops the nucleus, the rough endoplasmic reticulum, the smooth ER, the Golgi apparatus, the vesicles, the ribosomes, the nucleolus, the lysosomes, the mitochondria, the chloroplasts, the cytoskeleton, the cell wall, and vacuoles. So let's take each of these one step at a time, and I'm going to say up front that I'm not going to use analogies that other people teaching AP Bio are going to use, like calling the mitochondria the powerhouse of the cell, which may be beneficial, but you're not going to be able to use that terminology on the AP exam anyway, so I'd rather have you understand the function in the cell and be able to describe it from your own understanding, rather than giving you an analogy that you could forget to not write about on the exam. Let's start with the first organelle I mentioned, the cell membrane. It regulates what enters and exits the cell by keeping the internal and external environment separate, and I'll get into a lot more of its functions in a minute. The nucleus protects and houses DNA and lets mRNA out of the pores of the nuclear envelope/membrane to make proteins. You probably have noticed that I've used the word mRNA twice now instead of RNA. And just note that mRNA is messenger RNA, meaning that it is the RNA that is sent out of the nucleus to the ribosome with a copy of sorts of the instructions on which protein to create from the DNA blueprint, or from the DNA instructions, from the DNA genetic code rather. The rough endoplasmic reticulum specializes in making proteins because it is covered with ribosomes, and the smooth endoplasmic reticulum is not covered with ribosomes, and rather synthesizes lipids right outside of the nucleus, including phospholipids for the very important cell membrane. And just a little extra note here, the smooth ER also helps to detoxify harmful compounds created within metabolism, or toxins that have entered the cell. And because the liver does this in our bodies, the liver has lots of smooth ER for detoxification in its cells. The Golgi apparatus modifies and folds proteins into secondary, tertiary, and quaternary structures and adds the necessary carbs, lipids, and other proteins to them to get them ready for whatever function they will be performing. And the vesicles transport chemicals or cellular components around within the cell. And ribosomes, which remember are not membrane-bound since they exist in prokaryotes, which have no membrane-bound organelles, make proteins from the mRNA instructions. And ribosomes are made of rRNA that the nucleolus creates from within the center of the nucleus. Lysosomes break down substances for recycling purposes and non-functioning organelles. And the mitochondria is the site of cellular respiration, which is a huge concept that we'll be discussing in Unit 3 a lot. Additionally, they aid in the process of programmed cell death, or apoptosis, that can happen if a cell has too many genetic mutations as it is trying to reproduce in mitosis, for example. Chloroplasts are the site of photosynthesis, which we'll also be discussing a lot in Unit 3, and the cytoskeleton gives structural support for cells. The cell wall gives plants and some bacteria rigidity and shape, which is why a plant feels firm when you touch it, and the vacuoles store water, ions, and food, and specifically the central vacuole in plants is actually larger than the nucleus and is full of water that keeps the plants firm. If a plant is nice and firm, it is said to be turgid or have a high turgor pressure, meaning that the central vacuole is full of water. Now that we've talked a lot about how the inner parts of the cell work, let's talk about specific differences between animal and plant cells, which I hinted at a second ago when talking about the cell wall and central vacuole. It is important to note that plant cells have a cell wall made of cellulose, have that large central vacuole, have chloroplasts where photosynthesis takes place, and usually have a similar rigid box-like structure. On the other hand, animal cells can have cilia and flagella that I mentioned earlier, which aid in movement, lysosomes, which aid in digesting and breaking down chemicals or non-functioning organelles. Oh, and I forgot to mention earlier that lysosomes also assist with apoptosis via releasing hydrolytic enzymes that break down cell components in cell death. But back to animal cells' unique characteristics, animal cells also uniquely consist of peroxisomes, which aid in waste degradation in animal cells, and centrioles, which aid in the structure of the cell. It is also important to note that animal cells differ from plant cells in their shape. While plant cells generally have the same box-like structure, animal cells usually have different forms and shapes. For example, muscle cells are shaped very differently from neurons, but are even more different from red blood cells. Okay, now let's zoom into one specific part of the cell that is very important in this unit, the cell membrane, which is made of phospholipids that I briefly discussed in the last video. Like I mentioned last time, phospholipids have a phosphate group and a polar glycerol group on its hydrophilic head, and it has hydrophobic tails, which can either be saturated or unsaturated. Unsaturated tails have double bonds and have kinks, while saturated tails are flat and have no double bonds that could cause kinks. And phospholipids as a whole are amphipathic, meaning that they have hydrophilic heads and hydrophobic tails. And because they are amphipathic, they form a bilayer, meaning that the polar heads are oriented toward the aqueous environments, while the hydrophilic tails are oriented away from aqueous environments. Now, to describe the cell membrane, we use a fluid mosaic model, with the fluid part of that term referring to the membrane being held together by weak hydrophobic interactions and can shift up and down, while the mosaic part refers to the cell membrane being comprised of many macromolecules, not just phospholipids, that we'll get to in a second. Going back to fluidity specifically, it's important to note that temperature greatly affects fluidity, and unsaturated hydrocarbon tails in particular help maintain fluidity at low temperatures because of their kinked tails, which prevent tight packing of phospholipids. Saturated hydrocarbon tails are flat, so they're not very useful to organisms living in lower temperatures because they have no kinks. Remember when I said in the last episode that it is important to note when an organism has saturated or unsaturated phospholipid tails? That's why organisms that live in cooler temperatures benefit from having an unsaturated tail in their phospholipid bilayer, while organisms that live in warmer temperatures may have saturated hydrocarbon tails, since the unsaturated ones don't benefit them in that environment. In addition to saturation of hydrocarbon tails affecting fluidity, the presence of cholesterol in the cell membrane also helps regulate fluidity at high and low temperatures. At high temperatures, the presence of cholesterol reduces the movement of phospholipids, while at low temperatures, the presence of cholesterol reduces tight packing of phospholipids at low temperatures. So, keep that in mind. Now let's embellish the mosaic part of the fluid mosaic model a little bit more. Macromolecules that exist in the cell membrane include membrane proteins, first of all, which consist of integral proteins and peripheral proteins. And do note that if a substance was trying to get across the cell membrane while attached to a certain protein, it wouldn't be able to cross the cell membrane if the receiving cell did not have a membrane receptor receptive to that particular molecule or protein. That's kind of a side note, but that's important to know since you may be thinking that as long as a substance can get through the transmembrane protein, it's into the cell, but that's not necessarily the case. Anyways, back to membrane proteins. Integral proteins are proteins embedded into the lipid bilayer and extend through the whole membrane, which is why they are also called transmembrane proteins, what I was just talking about, since they go through the whole membrane. And a key example of an integral protein would be an aquaporin. And if a protein like an aquaporin is mutated on the cell membrane, you'll see that that can cause some huge issues in terms of the function of the whole organism. However, peripheral proteins are a little bit different. They are not embedded into the lipid bilayer. Rather, they are loosely bonded to its surface and either exist on the top or on the bottom of the bilayer. In addition to membrane proteins, there are also membrane carbohydrates that you should know are important for cell-to-cell recognition. First, glycolipids are carbohydrates bonded to phospholipids, and glycoproteins are carbohydrates bonded to proteins, and the latter is the most abundant membrane carbohydrate. An important example of a glycoprotein is an ID tag like an antigen, which is used to determine whether a cell is safe or foreign, and blood cells have these for identification purposes, and they are very important in the immune system. Okay. Kind of like I mentioned before, substances can only enter the cell if they meet certain qualifications. Building on that is the idea of selective permeability, which encapsulates what I just said. Elements can either have an easy or hard time crossing the membrane. Molecules that have easy passage across the membrane include small, nonpolar, hydrophobic molecules, whose receptors are in the nucleus or cytosol, which is the fluid within the cytoplasm. And examples of these molecules with easy passage include hydrocarbons and small gases like carbon dioxide, diatomic oxygen, and diatomic nitrogen. You may be thinking, what makes them so special? Well, the reason why nonpolar molecules like the ones I just mentioned and even hormones like testosterone can cross the membrane unassisted is because the bulk of the membrane is hydrophobic. The bulk of the cell membrane, is hydrophobic. The hydrophobic tails. So it's not like there's a huge polar section that these molecules must cross. Now, the elements with difficult passage across the membrane are those polar hydrophilic molecules, the large molecules, and ions. And examples of these substances that absolutely need a membrane protein to cross the membrane are sugars, large amounts of water, and ions like sodium chloride. So how do we get these substances across the membrane? There are two ways, via passive transport or active transport. Let's start with passive transport, since it requires the least amount of energy. In fact, none at all. Passive transport is the transport of a molecule that does not require energy from the cell, because a solute is moving with its concentration or electrochemical gradient, from a high concentration to a low concentration. And that's how all molecules move, actually. Substances move from a high to low concentration of that substance naturally. They move down the concentration gradient, and this principle is called diffusion. When a molecule is moving from a high to low concentration of itself, it diffuses directly across the membrane. However, it is important to note that two factors in particular affect the rate of diffusion: the concentration of the solute and the temperature. If you have more solute, the higher the rate of diffusion, since more of that substance spreads out, and then molecules spread faster at higher temperatures, so the rate of diffusion increases as temperature increases. Of course, there are different rates of diffusion for different molecules, and even with diffusion, the membrane is still selectively permeable, which is related to the topic of osmosis. Osmosis is the diffusion of water down its concentration gradient across a selectively permeable membrane. So again, molecules travel from a high concentration to a low concentration of themselves, but it is important to note that water's movement can be thought of in two ways: traveling from a high to low concentration of itself, or traveling from a low solute concentration towards a high solute concentration. In other words, if there is a lot of salt, for example, outside of the cell, the water inside the cell is going to move towards the solute, since it's the water that is moving from a low to high concentration of salt, not the salt moving from a low to high concentration of itself. We'll talk more about that later with hypotonic versus hypertonic cells, so stay tuned. Anyways, back to passive transport. So basically, there are multiple ways that passive transport is manifested in a cell. The main way is via facilitated diffusion, or the diffusion of molecules through the membrane via transport proteins. These transport proteins are really helpful because they increase the rate of diffusion for small ions, lots of water, and carbohydrates. Within transport proteins, you can either have channel or carrier proteins, and each transport protein is specific for substances that it can facilitate movement for. So for example, the aquaporins that I just mentioned earlier specifically work with water, and a membrane protein will be specialized in that it only facilitates transport for a certain type of molecule, like a certain type of ion. It is important to note that facilitated diffusion once again is passive transport because the substance that moves across the membrane via facilitated diffusion is still moving down its concentration gradient with no energy needed. So back to the specific types of transport proteins, let's start with channel proteins, which are like a hallway in that they provide a channel through which molecules and ions can pass. And this channel is hydrophilic since it allows ions and polar molecules to pass through it. Many channel proteins are actually gated channels that only allow passage when there is a stimulus, and the most common example of this is what I was talking about earlier, the aquaporin, which helps transport large quantities of water across a cell membrane. Aquaporins are very important channel proteins because they regulate the amounts of water in certain parts of the body, such as in the kidneys. If aquaporins were not functional as a result of a mutation that could have altered the amino acid sequence, a pH change could cause the aquaporins to malfunction, or a temperature change, since membrane proteins have an optimal temperature, could cause these aquaporins to malfunction. And in this case, water would still be able to cross, but it would have a much lower rate of diffusion. However, it is important to note that other factors can cause a difficulty in the aquaporins being able to regulate the amount of water present in certain areas of the body. For example, if a lot of cisplatin is present in the body as a result of perhaps cancer treatment, more water is in the urine of a patient since the kidneys' aquaporins are having a hard time absorbing and transporting the water before it is excreted. Next, we have carrier proteins, which push substances through the cell membrane. They undergo a conformational change, or a change in shape, for a substance to pass through them, but still keep in mind that no energy is required to make a conformational change take place because we are still talking about passive transport. Examples of molecules that require a carrier protein include sugars and amino acids. Okay, we've talked a lot about passive transport, but what about active transport? Well, active transport is the transport of a molecule that requires energy because it moves a solute against its concentration gradient. And keep in mind that the only time active transport is needed is when a molecule is going against its concentration gradient, or in the case of endo or exocytosis, which indicates that the molecule is way too large to cross the cell membrane without energy. Types of active transport are pumps, cotransport, exocytosis, and endocytosis. So you keep on hearing me say energy is or is not required. Well, what do I mean by energy? I'm so glad you asked. When I say energy, I'm talking about ATP, or adenosine triphosphate, which is the usable energy source used by cells. ATP can transfer its terminal phosphate group to the transport protein, which changes the shape of the transport protein to better move a substance in a process called ATP phosphorylation. And again, this process of changing the shape of the transport protein is called a conformational change. Now you may be thinking, wait, Belladonna, you said a minute ago that carrier proteins undergo conformational changes in order to better transport substances across the membrane, but now you're saying that that requires ATP. But I thought passive transport didn't require energy. Good question. So basically, yes, carrier proteins do require conformational changes to transport substances from a high concentration to a low concentration of that substance. But the conformational changes that carrier proteins undergo in passive transport to transport a substance down its concentration gradient occur naturally and actually do not require ATP. It is only the conformational changes in active transport, in which a substance needs to be transported against its concentration gradient, that ATP phosphorylation is necessary to induce a conformational change on a transport protein. Anyways, now let's actually talk about the types of active transport, starting with pumps. Pumps maintain membrane potential by gathering positive charges and pumping them to one side, and membrane potential describes the unequal concentrations of ions across the membrane and how it results in an electrical charge, or electrochemical gradient. In this scenario where pumps are used, the cytoplasm is relatively negative in comparison to the extracellular fluid. And to get a little more specific, let's talk about electrogenic pumps, which are proteins that generate voltage across membranes by pumping positives or negatives across the membrane, which stores energy and creates a kind of slingshot effect when the energy is released. Specifically, in the case of a sodium-potassium pump, animal cells regulate their concentrations of sodium cations and potassium cations. When ATP phosphorylates, it bonds with the membrane protein, giving up its third phosphate group so that the sodium cation can be pumped out of the cell. When the potassium cation comes into the cell, the third phosphate group is released and the cycle starts all over again. So that makes a little more sense, let's use the example of neurons firing. In order to fire, neurons use the sodium-potassium pump to pump 3 sodium cations out of the nerve cell, while 2 potassium cations are getting pumped into the nerve cell, which results in a positive net charge in the extracellular fluid, since positive charges are being pumped outside of the cell. Through pumping these ions across the membrane in ways that act against their respective concentration gradients, energy is being stored, and a strong electrochemical gradient is being created. If the electrochemical gradient is strong enough, it will trigger an action potential, a brief electrical impulse created by the movement of ions that causes a neuron to fire. Once the action potential is generated, the membrane proteins of the axon, a part of the neuron, open, and the sodium ions that were being pushed out rush into the cell, while the potassium that was pushed in rushes out of the cell. And through the charge of the neuron changing briefly from negative 70 millivolts to positive 30 millivolts, an electrical impulse was created that caused the neuron to fire. While I would not think that you're required to know that specific example, I think it helps to explain the concept through the example, you know? Okay, now that we've talked about electrochemical or electrogenic pumps, let's talk about proton pumps, which are integral membrane proteins that build up a proton gradient across the cell membrane. It is used by plants, fungi, and bacteria, and they pump H+ ions out of the cell so that an electrochemical gradient created from doing that powers the movement of another ion to go against the gradient. And this leads to the idea of co-transport, which is another type of active transport that uses a proton pump to couple a favorable movement of one substance with an unfavorable movement of another substance. Co-transport uses the energy stored in the electrochemical gradient by those handy-dandy proton pumps to move substances against their concentration gradient. And just so we're clear, a favorable movement means that something is going down from high concentration to a low concentration, a normal concentration gradient, while an unfavorable movement is going from a low concentration to a high concentration, against the normal concentration gradient. Plants in particular use co-transport for sugars and amino acid transportation. And a great example of co-transport in action is the sucrose H+ co-transporter. Sucrose is a pretty hefty molecule, so it needs some help to go inside the plant cell. In reality, sucrose can travel into a plant cell against its concentration gradient only if it is coupled with hydrogen plus ions that are diffusing down their electrochemical gradients. In reality, sucrose can travel into a plant cell against its concentration gradient only if it is coupled with H+ that is diffusing down its electrochemical gradient. Keep in mind that a lower pH is more acidic, and a low acidity means that whatever solution or environment has that low acidity is high in hydrogen plus or H+ ions. So sucrose uptake would increase because because of that plethora of H+ ions for co-transport. So just keep that in mind, that certain environmental conditions can certainly facilitate processes like co-transport in plants. Now we are on to the last types of active transport, and you heard me say its opposite, endocytosis, a lot earlier, so let's define them both now. Exocytosis and endocytosis make a way for large molecules to be transported across the cell membrane. And because of these molecules' large size, energy is required. First, exocytosis is the secretion of molecules via vesicles that fuse to the plasma membrane, and the vesicles fuse to the membrane by forming a bilayer, and then release their contents into the extracellular fluid once it fuses to the cell membrane. Going back to neurons, and by the way, you can probably tell I love psychology since I keep bringing them up. A great example of how neurons use exocytosis is when they release neurotransmitters like acetylcholine, dopamine, serotonin, endorphins, and GABA into the synaptic cleft or tiny space between two neurons. On the other hand, endocytosis is the uptake of molecules from vesicles fused from the plasma membrane, so it's the opposite of exocytosis. The three main types of endocytosis are as follows: phagocytosis, pinocytosis, and receptor-mediated endocytosis. Phagocytosis is kind of like cell eating, since it is when a cell engulfs particles like macromolecules to be later digested by lysosomes. The way that this works is that the cell surrounds the particles with pseudopodia, which according to Google is when a cell temporarily protrudes out from its normal size to engulf particles in the eating process. Then it packages particles into a vesicle, and those particles are then transferred to a food vacuole, which then fuses with a lysosome to be digested. Pinocytosis is kind of like cell drinking, and it is the nonspecific uptake of extracellular fluid containing dissolved molecules. The cell takes in dissolved molecules in a protein-coated vesicle, and it has a protein coat to help mediate the transport of molecules. Then lastly, we have receptor-mediated endocytosis, which is the specific uptake of molecules via solute binding to receptors on the plasma membrane. And when the solutes bind to the receptors, they cluster in a coated vesicle to be taken into the cell. Remember when I said earlier that a substance attached to a protein wouldn't be able to cross if the receiving cell did not have a membrane receptor receptive to that protein? Receptor-mediated endocytosis is kind of like that, and it allows the cell to take up large quantities of a specific substance. Okay, now that we've covered passive and active transport, let's go back to osmosis and diffusion for a little while and explore the ideas of hypotonic versus hypertonic cells through osmoregulation and tonicity. Osmoregulation describes the fact that cells must be able to regulate solute concentrations and maintain water balance, otherwise things could go haywire. Building on that, tonicity is the ability of an extracellular solution to cause a cell to gain or lose water, and it depends on the concentration of solutes that cannot pass through the cell membrane. So if the concentration of non-penetrating solutes is higher outside of the cell, the water will move out of the cell and into the extracellular fluid, since I said earlier that water moves from a low to high solute concentration and high to low water concentration. Naturally, if there is higher solute concentration outside the cell, there are slightly fewer free water molecules moving around that are not binded to a solute, so this makes sense. Unfortunately, if water is moving outside of a cell due to a high salt concentration in the extracellular fluid, the cell will eventually shrivel and die, and a cell that is in this situation is in a hypertonic solution, meaning that the cell loses water due to its extracellular surroundings. Plant cells specifically, being immersed in a hypertonic solution causes their central vacuole to shrink and the plasma membrane to pull away from the cell wall, which is detrimental to the health of the cell and plant as a whole and is known as plasmolysis. On the other hand, if the concentration of non-penetrating solutes is higher on the inside of the cell than the outside, the cell will gain water, since the water will move from a high to low water concentration or a low to high solute concentration, which will cause animal cells to swell and lyse or burst from all the water inside the cell. But plant cells work optimally, because lots of water entering the cell is a positive thing, because that great amount of water will be stored in their central vacuole and result in a high turgor pressure, making the plant rigid and firm as a whole. Okay, we are almost done with episode 2 of That Bio Girl, but I said earlier that we would talk about cell size and water potential, so here we go. So since cells produce waste, dissipate thermal energy, and need to exchange resources like chemical nutrients across the cell membrane, cells need a higher surface area to volume ratio to optimize the exchange of materials through the plasma membrane. Smaller cells are more efficient in performing chemical reactions more quickly because of the higher surface area to volume ratio, which is great for chemical reactions and their efficiency. The reason why we humans have so many small cells instead of a few big cells is because bigger cells are less efficient, even though they are great for storage. Now, if you're wondering about how eukaryotic cells and prokaryotic cells compare in terms of productivity, since eukaryotic cells are bigger than prokaryotes, you should give yourself a pat on the back because that's a great question. While eukaryotic cells are indeed bigger than prokaryotes, they are more efficient than prokaryotes because of what I talked about earlier, compartmentalization. Because they have organelles like the mitochondria and chloroplasts that consist of folds that add to the surface area of the eukaryotic cell as a whole, there is more room for chemical reactions to take place, while not having to increase the cell's size, making the cell very efficient. A good example of a cell that is very efficient is a muscle cell, which has a lot of mitochondria that have lots of folds to create energy when you're walking or exercising. Hence, the large amount of mitochondria contributes to a more efficient cell. Also, I should mention that if you are asked to determine which cell is more efficient, which is likely, look for the smallest cell or the one with the highest surface area to volume ratio. And if you need a refresher on how to calculate or find that, just look on the AP Biology reference sheet, and it should be on there in the surface area and volume section. Okay, now we are on the last section of this episode, water potential. It may sound scary, but don't worry, it's okay. Water potential is a physical property that predicts the direction in which water will flow, and it takes the effects of solute concentration and physical pressure into consideration. In terms of water potential, water will flow from areas of high water potential to low water potential, and it will flow from a low solute concentration to areas of high solute concentration, and from high pressure to areas of low pressure. Now, you will see this in your course, but the water potential formula is the Greek letter psi, or psi, it's psi, you could say it both ways, is equal to the solute potential plus the pressure potential. Let's break each of those symbols down, starting with psi. I'm going to call it psi. Psi is measured in megapascals, MPa, or bars, and just so you know, 1 megapascal is equal to 10 bars. Next, solute potential is also known as osmotic potential, and it is important to note that an increase in solute leads to a decrease in free water molecules, because if you think about it, adding salt, for example, to a solution will mean that more water molecules bind to the extra salt molecules, thus decreasing the number of free water molecules and thus decreasing water potential. Solute potential is usually expressed as a negative number, and pure water has a solute potential of 0 megapascals and an overall water potential of 0 megapascals or 0 bars. You're probably wondering, how do I calculate solute potential? Great question. The solute potential is equal to the negative value of ICRT, with I equaling the ionization constant, C equaling the molar concentration, R equaling the pressure constant, and T equaling the temperature in Kelvin. Now let's break down each of those parts. The ionization constant is the number of particles formed. So for sodium chloride, NaCl, the ionization constant would be 2. And if no ions are formed, as in the case of sucrose, the ionization constant would just be equal to 1. The molar concentration describes the molarity of the solute, or how many moles of the solute are present per volume of solution. And also note that similar to what I said earlier, adding more solutes leads to a more negative solute potential because adding more solute decreases the number of free water molecules, reducing the tendency of the water to move. So in other words, leading to a more negative and lower water potential. The pressure constant is a constant value that you use depending on whether the pressure is in megapascals or bars. If the situation calls for bars, use 0.0831 liter bars per mole Kelvin. And if the situation calls for megapascals, you use 0.00831 liters megapascals per mole Kelvin. And then lastly, with regards to temperature, you must use a temperature in Kelvin, which is just 273 plus your temperature in Celsius. Don't forget this step, because using a Celsius temperature instead of a Kelvin one will give you the wrong solute potential, and thus the wrong water potential. Lastly, we have pressure potential, the physical pressure on a solution. It can either be positive or negative, but it is relative to the atmospheric pressure, so open air would give you a pressure potential of 0 megapascals, since no outside forces would be creating pressure as they would be in a closed container. In this case, since the pressure potential is 0 in an open container, since the pressure potential is 0 megapascals or 0 bars in an open container, the solute potential is equal to the water potential. Now, to be clear, your water potential values can either be negative, 0, or positive, depending on your values, but it is very important to note that the more negative water potential you have, the more likely water will move into that area with negative water potential. So now I want to talk about some key ideas that you should memorize in relation to water potential and how increasing and decreasing solute concentration will affect it, from College Board's AP Classroom Unit 2.8 Daily Video 3. First, this video states that increasing the amount of solute in water will cause an increase in solute potential and a decrease in water potential, which makes sense because we said earlier that water diffuses from a low solute concentration to a higher solute concentration, and water moves from areas of high water potential with less solute to areas of lower water potential with more solute. Similarly, decreasing solute concentration will result in a decrease in solute potential and an increase in water potential, which again makes sense since water with less solute or higher water potential will still diffuse from areas of low solute concentration to areas of high solute concentration, since areas of low solute concentration have more free water molecules and areas of high solute concentration have fewer free water molecules, thus allowing water molecules to travel from high water concentration to areas of low water concentration, or areas of high water potential to areas of low water potential. Other important things to note are that increasing water potential will cause an increase in pressure potential, and decreasing pressure potential will cause a decrease in water potential. Okay, so basically, just to recap, water potential is used to answer the question, how likely is the water to move based on pressure and the solute? Or how likely is the water to move by osmosis? So a great application of water potential is to determine which way water will move in a plant, from the roots to the leaves or from the leaves to the roots, from the leaves to the air or from the air to the leaves. So using the question from AP Classroom Unit 2.8 Daily Video 3, specifically from Kelsey Burris, let's work through this question together. And just to be clear, you will have to know how to calculate water potential for the AP Bio exam, so please practice that if you're going to be taking that exam. But for this podcast, I'm more focused on helping you understand the concept and how it might look in a practice question that tests your conceptual knowledge of water potential, as opposed to teaching the calculations. So let's look at Mr. Burris's practice question. A new tree is planted in a soil with a water potential of -4 bars. The roots of the tree have a water potential of -8 bars. Which way will the water move? Okay, so we already know a few things here. First, we can assume that the roots of the tree have a higher solute concentration and thus fewer free water molecules than the soil. And we also know that the -4 bars in comparison tell us that the soil has more free water molecules and a lower solute concentration than the roots because it has a higher, less negative water potential. So because the soil's water potential of -4 bars is more positive, or less negative, than the root's water potential of -8 bars, we can safely state that the soil has a higher water potential, or tendency to move, than the roots. And the root's water potential of -8 bars shows us that the roots have a lower water potential since it is a more negative value. Therefore, since we know that water moves from areas of higher water potential to areas of lower water potential, or areas of higher water concentration to areas of lower water concentration, or areas of lower solute concentration to areas of higher solute concentration, we can confidently state that the water will move from the soil soil with the -4 bars water potential to the roots with the -8 bars water potential. Now, what if I told you that the leaves had a water potential of -17 bars? Which way will the water move now? From the roots to the leaves or from the leaves to the roots? Using the same principle, we can state that the water will move from the less negative water potential of -8 bars, in this case in the roots, up the tree into the leaves whose water potential is more negative and thus lower than -8 bars, -17 bars. Now you may ask, why do the leaves have a higher solute concentration and a lower water potential than the roots? Well, keep in mind that the roots often store water for an organism like a tree, so roots have a higher amount of free water molecules than the leaves because of the roots' storage function, which means that they have a higher water potential than the leaves and will move into the leaves due to their more negative water potential. Now, as for leaves, you might say that they have a higher solute concentration because leaves are where photosynthesis takes place, and in photosynthesis, glucose is created. So, in that case, glucose would be present more in the leaves than in the roots, and thus, fewer free-moving water molecules would be present in the leaves. So now what if I told you that the atmosphere's water potential was -1,152 bars? Where would the water move then? Well, the water would move from the leaves into the atmosphere since the more positive water potential of -17 bars would move towards the lower water potential of -1,152 bars via evaporation. Okay, so I know that was a lot about water potential, but just some key takeaways that I liked from College Board's AP Bio Daily Video 3 under Section 2.8 are the following. Number 1, water potential moves by osmosis from areas of high water potential, or tendency to move, to areas of lower water potential. 2, water moves by osmosis from areas of low solute potential to areas of high solute potential. And 3, osmoregulation, which water potential is part of, maintains water balance and allows organisms to control their internal solute composition and water potential. So I know that I was repeating a lot of important facts here, and you may be wondering, why does Belladonna keep on repeating the things that she said over and over again? Well, repetition is very important because it emphasizes how important an idea is, and you absolutely cannot forget the facets of water potential. So I like repeating them to make sure that you will not forget them. Okay guys, I know that was a lot of content, but you got through it, and as always, I hope that this second episode of That Bio Girl strengthened your understanding of the processes in Unit 2 as a whole, and stirred your appreciation for the biological world around us. If you liked this episode, feel free to like and follow That Bio Girl on Spotify, Instagram, Amazon Music, and Audible. Tell your friends about it, and keep on dreaming. See you next time!